most of the usable energy obtained from the breakdown of carbohydrates or fats is derived by oxidative phosphorylation, which takes place within mitochondria along with the final and the most important step of cellular respiration, the electron transport chain. While glycolysis and the citric acid cycle make the necessary precursors, the electron transport chain is where a majority of the ATP is created to suffice the need of energy to survive. Electron transport chain is a sequence of complexes found in mitochondrial membrane that uses reducing equivalents to generate more ATP. The breakdown of glucose by glycolysis, oxidation of pyruvate to acetyl coenzyme A, and the citric acid cycle yields a total of 4 molecules of ATP, 10 molecules of NADH, and 2 molecules of FADH2. Electrons from NADH and FADH2 are then shuttled across the mitochondrial membranes, producing an electrochemical gradient, and through proton driven chemistry of the ATP synthase molecule, electrons get transferred to molecular oxygen, generating ATP, the adenosine triphosphate molecule, by oxidative phosphorylation. The discovery in 1948 by Eugene Kennedy and Albert Leninger that mitochondria are the site of oxidative phosphorylation in eukaryotes marked the beginning of the modern phase of studies in biological energy transductions. Mitochondria have two membranes, outer and inner. The inner membrane is impermeable to most small molecules and ions, including protons. Only species that do so through specific transporters. The inner membrane bears the components of the respiratory chain and the ATP synthase. These components are NADH coenzyme Q oxidoreductase, which is complex 1, succinate Q oxidoreductase, which is complex 2, Q cytochrome C oxidoreductase, that is complex 3, and cytochrome C oxidase, which is complex 4, along with mobile electron carriers like ubiquinone Q and cytochrome C, which serves as links between ETC complexes. All proteins within the chain move back and forth between the reduced and oxidized states as the electrons are passed to and from them. Now, we shall see these components in detail. Firstly, the NADH coenzyme Q oxidoreductase complex 1. Complex 1 is composed of flavin mononucleotide FMN and an enzyme containing iron sulfur cluster FES. FMN, which is derived from vitamin B2, also called riboflavin, is one of the several prosthetic groups or cofactors in the electron transport chain. The enzyme in complex 1 is NADH dehydrogenase, a very large protein containing 45 amino acid chains. Complex 1 can pump 4 hydrogen ions across the membrane from the matrix into the intermembrane space. In this way that the hydrogen ion gradient is established and maintained between the two compartments separated by the inner mitochondrial membrane. Next is succinate Q oxidoreductase complex 2. Complex 2 directly receives FADH2, which does not pass through the complex 1. The compound connecting the first and second complexes to the third is ubiquinone Q. The Q molecule is lipid soluble and freely moves through the hydrophobic core of the membrane. Once it is reduced to QH2, ubiquinone delivers its electrons to the next complex in the electron transport chain. Q receives the electrons derived from NADH from complex 1 and the electrons derived from FADH2 from complex 2, including succinate dehydrogenase. This enzyme and FADH2 form a small complex that delivers electrons directly to the electron transport chain, bypassing the first complex. Since these electrons bypass and thus do not energize the proton pump in the first complex, fewer ATP molecules are made from the FADH2 electrons. The number of ATP molecules ultimately obtained is directly proportional to the number of protons pumped across the inner mitochondrial membrane. Next is Q cytochrome C oxidoreductase, which is complex 3. The third complex is composed of cytochrome B, another FES protein, Rescue center, 2Fe, 2S center and cytochrome C proteins. This complex is also called cytochrome oxidoreductase. Cytochrome proteins have a prosthetic heme group. The heme molecule is similar to the heme in hemoglobin, but it carries electrons, not oxygen. As a result, the iron ion at its core is reduced and oxidized as it passes the electrons, fluctuating between different oxidation states, Fe2+ reduced and Fe3+ oxidized. The heme molecules in the cytochromes have slightly different characteristics due to the effect of the different protein binding them, which make each complex. Complex 3 pumps protons through the membrane and passes its electrons to cytochrome C for transport to the fourth complex of protein and enzymes. Cytochrome C is the acceptor of electrons from Q. However, whereas Q carries pairs of electrons, cytochrome C can accept only one at a time. Now, cytochrome C oxidase, which is complex 4. The fourth complex is composed of cytochrome proteins C, A and A3. This complex contains two heme groups, one in each of the cytochromes A and A3, and 3 copper ions, a pair of CuA and 1 CuB in cytochrome A3. The cytochromes hold an oxygen molecule very tightly between the iron and copper ions until the oxygen is completely reduced. The reduced oxygen then picks up 2 hydrogen ions from the surrounding medium to produce water. The removal of the hydrogen ions from the system also contributes to the ion gradient used in the process of chemiosmosis. The net reaction is 4 cytochrome C Fe2 plus 4H plus O2 gives 4 cytochrome C Fe3 plus 2H2O. Ubiquinone Q. It is a membrane soluble low molecular weight compound, also called as coenzyme Q. It has long hydrophobic tail which keeps Q anchored into the mitochondrial inner membrane. Q is a lipid soluble molecule that diffuses within the lipid bilayer and shuttles electrons from complex 1 and 2 and pass them to complex 3. It is not a part of any complex. Now, the second one is cytochrome C. It is a peripheral membrane protein associated with the outer face of the membrane, which transports electrons and protons from complex 3 to complex 4. Cytochromes are heme containing proteins which contains iron. It is also not a part of any complex. First is NADH oxidation by complex 1. Each NADH transfers its 2 high energy electrons to FMN, then to an iron sulfur protein and finally to coenzyme Q, ubiquinone. NADH first binds to NADH Q reductase complex 1 and is reoxidized, leaving NAD plus. 2 electrons and 2 protons H plus are removed from NADH and transferred to the flavin mononucleotide FMN prosthetic group of NAD Q reductase to produce FMNH2. Electrons and protons are then passed to iron sulfur clusters in iron sulfur proteins or non-heme iron proteins within the complex, which change from Fe3 plus ferric state to Fe2 plus ferrous state when accepting an electron. The reaction catalyzed by complex 1 is NADH plus H plus coenzyme Q plus 4H plus gives NAD plus coenzyme QH2 plus 4H plus. Complex 1 to complex 3. Electrons and protons are transferred from NADH Q reductase to ubiquinone to form ubiquinol. It is small and hydrophobic and may therefore diffuse freely through the inner mitochondrial lipid bilayer, passing electrons and protons between the less mobile protein complexes. Electrons are then transferred to the next protein complex, cytochrome BC1, which is complex 3, which contains cytochrome B, cytochrome C1 and a risque FES protein. Cytochrome proteins contain a heme group that can bind an electron. Since cytochromes can only accept one electron, the 2 electrons from ubiquinol are transferred sequentially via the Q cycle. The net reaction is QH2 plus 2 cytochrome C Fe3 plus 2H plus gives Q plus 2 cytochrome C Fe2 plus 4H plus. Now the complex 3 to complex 4. Cytochrome C is a peripheral membrane protein, loosely bound to the inner mitochondrial membrane. It first binds to cytochrome BC1, which is complex 3, and accepts an electron from cytochrome C1 and transfers this electron to the next complex in the chain, cytochrome C oxidase, which is complex 4. The copper A CuA center of cytochrome C oxidase receives electrons one at a time from cytochrome C, which are then passed on to heme A and then heme A3. Oxygen binds to heme A3 and is reduced to water by the transfer of electrons and protons. Electrons are passed spontaneously along the electron transport chain in one direction due to increasing redox potential from NADH, most negative, to oxygen, most positive. However, many of the electron carriers in the chain utilize iron. Small distortions in the iron orbitals by the functional groups of the protein surrounding the iron atom give each subsequent carrier in the chain a higher redox potential. The net reaction is 4Fe2 plus cytochrome C plus 8H plus O2, which gives 4Fe3 plus cytochrome C plus 2H2O plus 4H plus. Now the generation of a proton electrochemical gradient. There is a large change in the redox potential at each of the 3 complexes: NADH dehydrogenase, cytochrome BC1 and cytochrome C oxidase, which corresponds to a large change in the free energy. This energy can be utilized to pump protons across the inner membrane from the mitochondrial matrix to the intermembrane space, creating a proton electrochemical gradient. NADH Q reductase and cytochrome BC1 each pump 4 protons, whereas cytochrome C oxidase pumps only 2. The protons then flow down their electrochemical gradient back into the mitochondrial matrix via an ATP synthase enzyme complex which generates ATP. Now, FADH2 oxidation by complex 2. FADH2 generated during the oxidation of succinate to fumarate by succinate dehydrogenase in TCA cycle transfers its electrons to coenzyme Q via complex 2. Electron transport from FADH2 does not involve the NADH Q reductase complex. Instead, electrons are transferred to the FES clusters of the succinate dehydrogenase enzyme, which also functions as a part of succinate Q reductase complex 2 of the electron transport chain. Electrons and protons are then passed on to ubiquinone and the rest of the complexes in the chain. The succinate Q reductase complex does not act as a proton pump, as the free energy change is insufficient to actively transport protons across the membrane. Oxidation of FADH2 therefore results in fewer protons being pumped across the inner mitochondrial membrane than NADH and produces less ATP. The net reaction is succinate plus coenzyme Q gives fumarate plus coenzyme QH2. The chemiosmotic hypothesis given by Peter D. Michel in 1961 suggested that the energy released during the electron transport chain is used to create a proton electrochemical gradient across the inner mitochondrial membrane, which can be used to generate ATP. Only 3 of the 4 mentioned electron carriers are capable of transporting protons from the matrix to the intermembrane space: 1, 3 and 4. It is this proton gradient that drives phosphorylation of ADP to ATP, as well as several other important transport systems. As proton concentration builds up in the intermembrane space, a gradient is created and protons are transported from high to low concentration. The energy from the transfer of protons is used to change ADP into ATP through phosphorylation. ATP synthase is the protein responsible for ADP phosphorylation. The enzyme ATP synthase embedded in the inner mitochondrial membrane allows the hydrogen ion H+ to flow back into the matrix through a hole in its center. It uses the ion's motion to convert ADP to ATP. This process is called oxidative phosphorylation because the ATP is phosphorylized using oxygen as the terminal electron acceptor. According to the IUBMB, International Union of Biochemistry and Molecular Biology enzyme nomenclature, the enzyme is called ATP phosphohydrolase. H plus transporting. However, the name ATP synthase reflects the primary function of the enzyme more clearly and nowadays is most widespread. ATP synthase is a large mushroom-shaped asymmetric protein complex. ATP synthase or complex 5 consists of two functional domains. F1 situated in the mitochondrial matrix, the cap of the mushroom, and F0 located in the inner mitochondrial membrane. These subunits are named by Greek letters alpha, beta, gamma, delta and epsilon in accordance with their molecular weight. F1 is composed of 3 copies of each of subunits alpha and beta, and 1 each of subunits gamma, delta and epsilon. F1 subunits gamma, delta and epsilon constitute the central stalk of complex 5. The proton translocating F0 portion is composed of subunits of 3 types named A, B and C. The function of ATP synthase is to synthesize ATP from ADP and inorganic phosphate in the F1 sector. This is possible due to energy derived from a gradient of protons which cross the inner mitochondrial membrane from the intermembrane space into the matrix through the F0 portion of the enzyme. The released energy causes rotation of the 2 rotary motors, the ring of C subunit in F0 relative to subunit A, along with subunits gamma, delta and epsilon in F1 to which it is attached. Protons pass F0 via subunit A to the C ring. Rotation of subunit gamma within the F1 alpha 3 beta 3 hexamer provides energy for ATP synthesis. In the case of ATP synthesis, each site switches cooperatively through conformations in which ADP and PI bind. ATP is formed and then released. ATP hydrolysis uses the same pathway. These transitions are caused by rotation of the gamma subunit. The alpha 3 beta 3 hexamer must remain fixed relative to the subunit A during catalysis. This occurs through the peripheral stalk. In oxidative phosphorylation, passage of 2 electrons from NADH to oxygen drives formation of about 2.5 ATP, and passage of 2 electrons from FADH2 to oxygen yields about 1.5 ATP. When both the pyruvate molecules are oxidized to 6 carbon dioxide via PDH complex and the citric acid cycle, and the electrons are transferred to oxygen via oxidative phosphorylation, as many as 32 ATP are obtained per glucose. Electron transport and oxidative phosphorylation are critical activities of protein complexes in the inner mitochondrial membrane, which ultimately serves as the major source of cellular energy. Around 95% of the ATP of the cell to survive is generated by these processes only.